Alaikum class. I hope you are all fine. I would like to welcome you all to the second phase of online teaching. And you all know that in this phase, you will be studying the fundamentals of genetics, which is a course which is offered by the Department of Biosciences. So the course code is Bio 231, and you all know that it's a 4 credit hour course in which 3 parts are for the theory and 1 part is for the lab. Assalamu alaikum class. I hope you're all fine. So today we start our studies on the fundamentals of genetics, in which we will be discussing today extensions and modifications of Mendel's laws. Before we start that, let's just quickly look at the course content that we have covered so far and the things that we are going to study today. So, the parts that we have covered are in green colored font, which include the study of life, which was about the brief introduction to biology and genetics, the chemical foundations of life, cell structure, cell division, mitosis, meiosis, cellular organelles. We have covered all these too previously. Then we have also studied about the biological membranes, their replication, transcription, translation, and types of RNAs. In our previous online lectures, we have started our series of lectures on Mendelian genetics, and I have divided that into 3 lectures, which are named as Mendelian Genetics Part 1 and Part 2 and Part 3, in which we have so far studied the law of segregation and dominance and the law of independent assortment. We have also done some examples and calculated the ratios using Punnett square and different type of crosses between different traits of the pea plants that Mendel used during his experiments. In today's lecture, we will be starting our next series of lecture on the extension and modification of Mendelian laws, which include terms such as incomplete dominance, complete dominance, multiple alleles, lethal alleles, epistasis, and the Bombay phenotype of the blood group. So let's get started. The extension and modification of Mendel's laws. We discussed the fundamental principle of transmission of genetics, and we saw that genes are present on homologous chromosomes and that these chromosomes segregate from each other and assort independently from other segregating chromosomes during gamete formation. Now, these two postulates are the basic principles of gene transmission from the parents to the offsprings. Once an offspring has received the total set of genes, it is the expression of the genes that determines the organism's phenotype. When gene expression does not adhere to a simple dominant and recessive mode, or when more than one pair of genes influences the expression of a single character, the classical 3 is to 1 ratio that was observed in Mendelian experiments and the 9 is to 3 is to 3 is to 1 F2 ratios are usually modified. In this video lecture and in several other lectures that we're going to do, we consider more complex modes of inheritance. In spite of the greater complexity of these situations and the fundamental principles which were actually set by Mendel still hold. In this lecture, we will restrict our initial discussion to the inheritance of traits which are controlled by only one set of genes, and in diploid organisms which have homologous pairs of chromosomes, 2 copies of each gene is going to, you will see that it's going to influence a certain trait. Now, before we actually start our lecture on these extensions of Mendelian's law, there are some terms that I have already explained, but I will be using these terms like again. So let's just briefly memorize them again. So first of all, we have allele. And you all know that allele is one of the number of alternative form of the same gene occupying a given position or locus on a chromosome. Then we have 2 types of alleles, either it's a wild type or it's a mutant type. And a wild type is the allele that like it's present in the nature and it's the it encodes the phenotype which is most common, which is occurring naturally, and that's called the wild type allele. Then we have the other type of the allele that's called mutant. Mutant is the type of allele which is not wild. And these alleles are like they can be found in a frequency lesser than the wild type of allele, and they're producing different phenotypic traits. Then we will be coming across the term of incomplete dominance, which is a condition in which the phenotype of the heterozygous genotype is distinct from and often intermediate to the phenotypes of the homozygous genotypes. Then another important term would be codominance, which is a condition in which both alleles of a gene pair in a heterozygote are fully expressed. Both of the alleles are showing their effect in the phenotype, with neither one being dominant or recessive to the other one. So these are the symbols for the genes, and you see that there are two systems for the symbol. We have Mendelian system and we have this newer system. Since Mendel worked with these that were either completely dominant or completely recessive, and there was initially he did not experience any intermediate traits, so he normally used for a dominant allele a capital letter, and a recessive allele was actually represented by a small letter. Here you can see that small a is actually representing a recessive trait, and capital A is actually representing a dominant trait or the dominant allele. Then we developed a newer system for representing the symbols of the genes. Here you can see in the newer system we have small letter or lowercase letter with a positive sign as a superscript. If it is representing a allele which is wild type and recessive, and a small letter a can simply represent a symbol of a mutant allele if it is recessive. And a dominant allele in the new system can be represented by a capital or uppercase letter A, along with the superscript, which may be a positive sign, or it can also be represented by just a positive sign with the superscript of the name of the allele, which is capital A, or just a positive sign. Similarly, the symbol for the mutant allele, which is the dominant mutant allele, can be represented by just a capital letter. For example, on the extreme right side in the base, you can see in the last row, it is being represented by a capital A or a uppercase letter, and it can be any letter, or a uppercase letter along with a positive sign as a superscript. So the things that we are going to study in the extensions and modification of Mendel's laws include incomplete dominance, codominance, multiple alleles, lethal alleles, and so on. The most that we are going to study today are incomplete dominance, codominance, and multiple alleles. Now, Greek and Mendel knew that how to keep things simple. In Mendel's work on pea plant, each gene came in just two different variations or alleles, and these alleles had a nice clear-cut dominance relationship with the dominant allele fully overriding the recessive allele to determine the plant's appearance. Today we know that not all alleles have quite a straightforward expression as in the Mendelian experiment. For example, in real life, alleles pair may have a variety of dominance relationship. That is, one allele of the pair may not be completely able to hide the other in the case of heterozygote. There are many different alleles for a gene in a population. In these cases, an organism's genotype or set of alleles set determine its phenotype or observable features. However, a variety of alleles may interact with one another in different ways to specify phenotype. As a side note, we are probably lucky that Mendel's genes did not show these complexities, and if they had, it's possible that Mendel would not have understood his results completely and wouldn't have figured out the core principles of inheritance, which are keys in helping us understanding these special cases. The learning objective of today's lecture is to discuss incomplete dominance, codominance, and multiple alleles as alternatives to dominance and recessiveness. So let's start with the first term, which is incomplete dominance. You all know that dominance describes the effect of different gene alleles on an organism's phenotype. An allele has complete dominance over the phenotype if one copy is sufficient to produce the same effect as two. An allele with incomplete dominance, however, will combine with the other allele to produce some intermediate phenotypes. Codominance is a special type of incomplete dominance when the heterozygous individuals simultaneously express both alleles or phenotypes equally. In general, a complete dominance refers to the heterozygous phenotype in is it's actually indistinguishable from the homozygous phenotype. When the heterozygotes express an intermediate phenotype that is distinct from the homozygous phenotype, incomplete dominance occurs. In incomplete dominance, the ratios of the genotype and the phenotypes are the same, but with complete dominance, they are actually different. Mendel's experiment with the pea plant suggested three main things. First, the two units or the genes or the alleles exist for every gene. Second, alleles maintain their integrity in each generation and they do not like blend. And number three is that the presence of a dominant allele, the recessive allele, is hidden and makes no contribution to the phenotype. Therefore, recessive alleles can be carried and not expressed by the individual. Such heterozygous individuals are sometimes referred to as carriers. Further genetic studies in other plants and animals have shown that much more complexity exists, but the functional principles of Mendelian genetics still hold true. Mendel results that the traits are inherited as dominant and recessive pair contradicted the view at that time that offsprings exhibited a blend of their parents' traits. However, the heterozygous phenotypes occasionally does not appear to be intermediate between the two parents. Here is a very simple example of incomplete dominance in plants, and we have this plant snapdragon, and here you can see a cross between a homozygous parents with white flower colors and a homozygous parent with red flowers, and when they are crossed together you can see in the F1 generation they are all producing pink flowers. When we look at the genes of these two true breeding parent plants, you can see that capital C, with a superscript of capital R or uppercase capital R are representing the alleles for the dominant red color. And in the white parent, you can see that the white color is being represented by the uppercase W. And when they are crossed together in the F1 generation, you see that the genotype of the pink flower plant is capital C with a superscript which is capital R, and the other allele is being represented by capital C with a superscript which is which is uppercase W. Now, this pattern of inheritance is described as incomplete dominance, and you can see that the F1 individuals they are neither white and they are nor red in color, and these are actually showing the expression of two contrasting alleles that such that the individual displays a intermediate phenotype. The allele for the red flower is incompletely dominant over the allele for the white flowers. However, the results of a heterozygote self-cross, as you can see on the right side, can still be predicted, just as Mendelian dominant and the recessive trait. And in this case, the genotypic ratio would be, as you can see in the Punnett scale that we have made here on the self-cross of these pink flowers, that we have one completely red plant, one completely white plant, and two plants that are producing completely pink flowers. So the ratio of red to pink to white is 1 is to 2 is to 1. Now let's look at the an example of incomplete dominance in case of higher organisms such as humans. Now, incomplete dominance is quite rare in humans. We are genetically complex and most of our traits come from multiple genes. However, there are few examples of incomplete dominance in humans as well. Incomplete dominance is just part of what makes our species so complicated and interesting. We have this common disease, not very common, a rare disease called Tay-Sachs disease, and it's an example of incomplete dominance in humans. Now, what happens in this disease is that a mutation in the gene coding for an enzyme, which is called hexoaminidase A, which is typically causes a neurological dysfunction. Now, this is because this enzyme normally breaks down the lipid byproducts called gangliosides in the cell's lysosomes. But when this enzyme is not present or it's not functioning properly, lipids build up in the brain and therefore they interfere with the normal biological processes. An individual who is homozygous for the mutant allele does not produce this enzyme, hexoaminidase, and therefore will suffer from this disease. However, an individual who is heterozygous and has one functional copy and one mutant copy, in that case, if you just look at the level of the hexoaminidase activity in those heterozygotes, which are also called the carriers now, they will have half of the normal level of this enzyme, which means that they have an intermediate phenotype which is arising from each allele's contribution or lack thereof. Since both the normal and the mutant alleles produce the same amount of protein or the enzyme, they are actually co-dominant and it's a type of incomplete dominance. Now we have co-dominant. A variation on incomplete dominance is called codominance, in which both alleles of the same characteristics are simultaneously expressed in the heterozygote. So an example of codominance in case of human is MN blood group. MN blood group is exactly the type of blood typing that we know, which is the ABO blood typing, but it is like not very well known to the common people. Now, since I told you that the process or phenomena of incomplete dominance and codominance is closely related, and in these both alleles are simultaneously expressed in the heterozygote. The MN blood group typing, which was actually identified by Karl Landsteiner and Philip Levine, it was discovered by them. They actually identified the presence of a glycoprotein molecule on the surface of the red blood cells. And that glycoprotein is actually a combination of carbohydrates and proteins, and it acts as a native antigen, providing biochemical and immunological identity to the individual. In this diagram, you can see three types of the blood groups, which may be M, Mn, and N. And on the right side, you see the red blood cells carrying either Mn, or only N glycoproteins on them and making them like different from each other. In case of human population, two forms of these glycoproteins actually exist, which can be like designated by M or N. An individual can either be M blood type or he can be like N blood type, and sometimes he can also be both of them. The MN system is under the control of a locus which is found on chromosome number 4, and it has the two alleles which are here written as L raised to capital M and L raised to capital N. Now, because humans are diploids, three combinations are possible, each resulting in a distinct blood type. Once again, the genotypic ratio can be 1 ratio 2 ratio 1, and it's exactly the same as Mendel has previously explained. Codominant inheritance is therefore characterized by distinct expression of the genes products of both of these alleles. This characteristic distinguishes codominance from incomplete dominance, where the heterozygotes express an intermediate, or we can also say a blended phenotype. For codominance to be started, both products must be phenotypically detectable. We are going to study another example of that in our later lectures, where we will examine the ABO blood type system. So now we have the multiple alleles. You know that the information stored in any genes is extensive and mutations can modify this information in many ways. Each gene produces a different alleles. Therefore, for any gene, the number of alleles within members of a population need not to be restricted to two. When three or more alleles of the same genes are present, we call them multiple alleles. Mendel implied that only two alleles, one dominant and one recessive, could exist for a given gene. We now know that this is an oversimplification. Although individual humans, which are all diploid organisms, can only have two alleles for a given gene, Multiple alleles may exist at the population level, said that many combinations of the alleles are observed. Note that when many alleles exist for the same gene, the convention is to be denoted with the same common phenotype or genotype among wild animal as the wild type. They are often abbreviated as a positive sign. Now, this is considered the standard or the norm, and all other phenotypes or the genotypes are considered to be the variants of this wild type or standard type, meaning that they deviate from the wild type. The variant may be recessive or it may also be dominant to the wild type allele. Here we are going to discuss the example of the multiple allele, which is the coat color in rabbit. You know, there are different type of rabbits which may be like brown in color. They may have like fur which is gray, or it can be like a little black. The Himalayan ones have black spots or patches in the white fur, and we have the albino ones which are completely white with like pink eyes. Now, all other phenotypes or genotypes, as I told you, there is going to be a wild one, and then there are going to be some variants. Now here, in the case of the coat color, in case of rabbits, we have four alleles that exist for this gene. We are going to call this gene C. As you can see in this diagram, that we have capital C, which is the wild type. Then we have small c, which is a variant, and the variant types can have like a plus sign on them, and you also see that some of them have a small h on them, and some of them have a small or lowercase ch on them. Now the wild type version is capital C, two capital C's with two superscripts, which is a positive sign, and they express a brown fur. Then we have the chinchilla phenotype, in which the variant gene is being represented by a small c, which has a superscript ch. It is expressed as a black tipped white fur. The Himalayan phenotype you see here, which are the white ones which have black patches on their fur, they are being represented by another variant form of this C gene, which is being represented by small c, and it has a superscript small h. And it has a black fur on the extremities and white fur elsewhere. Finally, we have the albinos or the colorless phenotype, which is being represented by the variant, which is small c, since it is a diploid organism, so it is being represented by two small c's, and it expresses a white fur. Now, in case of multiple alleles, dominance hierarchies can exist. In this case, the wild type allele is dominant over all other. Chinchilla is incompletely dominant over Himalayan and albino, and Himalayan is dominant over the albino. This hierarchy or allelic series was revealed by observing the phenotypes of each possible heterozygote offsprings by a number of studies. A very good example of multiple alleles in case of human is the ABO blood group. The simplest case of multiple allele occurs when the three alternative alleles of one gene exist. This situation is illustrated in the inheritance of ABO blood groups in humans, which was actually discovered by Carl Steiner in the early 1900s. Like as I told you about the MN blood type, the ABO blood system, exactly like the MN blood type, it is characterized by the presence of an antigen, you also call it protein, on the surface of the red blood cells. In case of the MN blood type, that was a glycoprotein, whereas in case of the ABO blood group, just remember it that it's a type of protein. Now, the A and B antigen are distinct from the MN antigen, and they are under the control of a different gene, and that gene is located on chromosome number 9. As in the case of MN system, one combination of the alleles in the ABO system exhibits a codominance mode of inheritance. The ABO phenotype of an individual is ascertained by mixing a blood sample with an antiserum which contains type A or type B antibodies. Now, when this antigen is mixed with the antibodies, if they react together, if they are actually present, it's going to cause clumping or agglutination of the red blood cells, and by which, by the process of this agglutination, somebody can know what is their blood group. When an individual is tested in this way, one of the four phenotypes can be revealed, as you can see on the checkerbox on the left side. Now, each corresponding antibody, as I told you, and each individual has either A antigen, which means that he has A phenotype or A blood group. And the antigen B, if it is present, it's going to be B phenotype, and the blood group is going to be type B. And if both of A and B antigens are present in the blood on the red blood cells, the phenotype is going to be AB, or the individual is going to have a blood group which is AB. And if both of these A and B antigens are not present on the red blood cells, the phenotype is going to be O, and the individual is going to have O blood group. In 1924, it was actually hypothesized that these phenotypes are inherited as a result of three alleles of a single gene. Now, this hypothesis was based on the studies of the blood types of many different families. Although different designations can be used, we actually use the symbol letter uppercase I with A uppercase, as you can see, and I with B or a small I to distinguish between these three alleles. Now, this uppercase letter I actually stands for the antigen which is present on the red blood cells. And that is actually the antigen. Another term which is used for the word antigen is isoagglutinogen. Now, if we assume that the IA and IB alleles are responsible for the production of the respective A and B antigen, and small I is an allele that does not produce that detectable antigen, we can list the various genotypic possible combinations and assign the appropriate phenotype to each, as you can see in this figure on the left side and on the right side. And in these, you can see that IA and IB alleles are dominant to the small I allele, but they are co-dominant to each other. Now, the biochemical basis of the ABO blood type system has now been carefully worked out, and we know a lot about it. The A and B antigens are actually carbohydrate groups, or carbohydrates are simply known as the sugars, and they are bound to the lipid or the fatty acid molecules which are protruding from the membrane of the red blood cells. You can see here these yellow lines are actually representing the membrane of the red blood cell, which is actually made of a lipid bilayer, and you see that some of the lipids are extending out on the outer surface, on the outer side of the red blood cells. And these hexagons that are attached with the help of a covalent bond are different units of the sugar molecule, in which the red ones are the glucose units. The pink ones, which have lines in them, are the unit of N-acetylglucosamine, which is also a sugar molecule, but it is modified. Then we have the greens ones, which are the galactose units, and then we have N-acetyl galactosamine, which are represented by the hexagons, which are green in color and have like lines inside them. Now, the specificity of the A and B antigen is based on the terminal sugar of the carbohydrate group. Terminal is the last one. Almost all individuals possess what is called the H substance to one or the two terminal sugars are added. In the figure, you can see that the H substance itself contains three sugar molecules, which is galactose, which is being represented by a you see here a green hexagon, and an acetylglucosamine and fucose molecule, and they are all linked together by the help of a covalent bond. In the case of a IA allele, which is responsible for an enzyme that can add the terminal sugar and acetylglucosamine to the H substance, whereas in case of the IB allele, it is responsible for a modified enzyme that cannot add an acetylglucosamine, but instead it can add a terminal galactose. Terminal is the last one. Now, heterozygotes, which have both IA and IB alleles, add either one or the other sugar at many sites, which are actually the substrates available on the surface of the red blood cells, which actually illustrate the biochemical basis of the codominance in individuals who are AB blood type. Finally, the persons who are of type O or have O type of blood group, and therefore the allele is small I raised to O, they cannot either add terminal sugar, which are actually added by the A and B, and these persons have only the H substance protruding from the surface of the red blood cell. So on the right side of the slide, you see in the green block, that is the precursor substance. And if the allele A is present in an individual, the blood group is type A. And on the H substance, N-acetylglucosamine units of the sugar are added. While if the individual is type B blood group, then on the terminal sugar that is added to the H substance is actually a galactose unit. And if the individual is AB, then both N-acetylglucosamine and galactose may be present on the H substance. While in the case of O blood type, since both of these units cannot be added to the terminal sugar, to the terminal side of the H group, it is just protruding out without any N-acetylglucosamine or galactose unit. Now, in 1952, a very unusual situation provided information concerning the genetic basis of the H substance. A woman in Bombay, she displayed a unique genetic history, which was inconsistent with her blood group. In need of transfusion, and when they checked her blood group, they found that both antigen A and B were not present, which means that her blood group was type O. But they came to know that her parents were either type A or type B, and she was also the mother of a kid who had a blood group type B, which means that genetically she was actually type B, but functionally she was type O. And this woman was subsequently shown to be like homozygous for a rare recessive mutation in the gene, which is called the FUT gene or FUT1 gene. And it actually encodes an enzyme which is called the fucosyl transferase. Now, this enzyme or this gene prevented her from synthesizing the complete H substance. In this mutation, the terminal portion of the carbohydrate chain protruding from the red blood cell membrane lacks the fucose, which normally is added to the enzyme. Now, in the absence of the fucose, the enzyme specified by the allele capital A or allele B apparently was unable to recognize the incomplete H substance as the proper substrate. Therefore, they were not able to add the N-acetylglucosamine or the galactose molecule to the H group. And therefore, she was like neither type A, neither type B. She was type O, and at the same time, the H group was also different from the one which is actually found in most of the population. Now, as a result, the ABO system genotype cannot be expressed in individuals which are homozygous for the mutant for this gene, which was FOT1 gene, even though they may have allele A or allele B, and neither antigen added in the cell surface, and they are functionally type O. And to distinguish them from the rest of the population, they are said to demonstrate the Bombay phenotype. The frequency of this mutation or this mutant FUT allele is exceedingly low, and hence the vast majority of the human population can actually synthesize the H substance in the right way. So the takeaways from today's lectures are: number one, incomplete dominance is the expression of two contrasting alleles such that the individual displays an intermediate phenotype. Codominance is a variation on incomplete dominance in which both alleles of the same characteristics are simultaneously expressed in the heterozygote. Diploid organism can only have two sets of allele for a given gene. However, multiple alleles may exist in the population level such that many combinations of the two alleles are observed. And then the complete dominance of a wild type phenotype over all other mutants often occurs as an effective dosage of a specific gene product, which means that the wild type allele supplies the correct amount of the gene product, whereas the mutant alleles cannot do that. And in the last, one mutant allele can also be dominant over all other phenotypes, including the wild type.